Good afternoon. I welcome the counselors and students of Diploma in Dairy Technology. Today we will be covering fundamentals of dairy microbiology for the course Milk Production and Quality of Milk. The contents covered in the presentation will be Introduction to Microbiology, Milk in relation to public health, factors affecting growth of microorganisms, and the last unit will be covering control of microbial spoilage. Unit 12 deals with Introduction to Microbiology. Since we are talking about microbiology in terms of milk, we will be talking about dairy microbiology. It can be defined as study of microorganisms found in milk and its products. When we talk about microorganisms, they can be classified as beneficial and harmful. The ones we say useful, they are mostly used in the desirable fermentations. Whereas the harmful ones, they can be the spoilage types and the pathogens which are capable of causing the human diseases. The common microorganisms which are found in milk are bacteria, fungi. Fungi consist of yeast and molds, viruses. All these microorganisms have been classified based on their morphology, biochemical characteristics and their genomic characteristics. We start with the bacteria. These are single-celled prokaryotes. The major types which are found in milk are: first, the lactic acid bacteria, which are also known as lab and related genera; coliform bacteria; third, the spore-forming bacteria; and lastly, Pseudomonas and related genera. So now we will take one of them stepwise. The most important group is of lactic acid bacteria and related genera. These are the normal flora of milk, facultative anaerobes, which are non-spore forming, gram-positive, and having catalase absent in them. This group consists of cocci and rods, which are capable of converting the lactose, that is the milk sugar, to lactic acid. This conversion takes place via two routes. One is homofermentation and other is heterofermentation. When we talk about the term homofermentation, means the major product is the lactic acid, whereas when we talk about heterofermentation, lactic acid along with other compounds, which are usually the aroma compounds, is produced. Most of the lactic acid bacteria are used as the starter cultures. Most common genera which are involved are Lactococcus, Streptococcus, Leuconostoc, Pediococcus, Lactobacillus, Propionibacteria, Bifidobacteria, and Brevibacteria. Since they are very important for dairy microbiology, all the students should be aware about their characteristic features. So first we take Lactococci. Lactococci were initially known as Streptococci and they were isolated from milk or cream. They occur singly in pairs or in chains as shown in the photograph. They are non-motile, as cocci they do not have a flagella, mesophilic in their temperature requirements, and homofermentative. The most common species is the Lactococcus lactis, which is used as a starter culture for dahi and some cheeses. Under the Lactococci, we have one more bacteria, that is Streptococcus thermophilus. How it is different from Lactococci? First, it occurs in very long chains. The chains can be from 18 to 20 cells long. It grows at 45 degrees C, and this microorganism is used as a starter for yogurt in association with Lactobacillus bulgaricus. The next is Leuconostoc. They are ellipsoidal in shape. They occur in pairs and chains. They are heterofermentative in nature. Therefore, they have the ability to form aroma compounds. The most common species which we have is Leuconostoc mesenteroides, subspecies Dextranica. Pediococcus is one more type of lactic acid bacteria which has a characteristic feature that it occurs in tetrad. Tetrad is a group of 4 cells which is capable of dividing into 2 planes. It is again a homofermentative bacteria, and the most common species is P. acid lacti. Lactobacilli. This is a large group of rod-shaped bacteria, and these rods can be very small to very long in shape. They are further subdivided into 3 groups that are Thermobacterium, Streptobacterium and Beta-bacterium. Lactobacillus is a characteristic example of Thermobacterium. Thermo means which loves growing at high temperature. Then you have Streptobacterium. The common example is Lactobacillus casei, and Lactobacillus brevis is example of Beta-bacterium. Propionibacterium is another type of lactic acid bacteria which is usually associated with Swiss cheese, as it is responsible for the characteristic eyes and flavor which is found in Swiss cheese. This is a non-spore forming anaerobic to aerotolerant bacteria, and it is gram-positive in nature. But instead of converting lactose to lactic acid, it converts it into propionic acid along with CO2, and this CO2 is responsible for the eye formation in the Swiss cheese. The species which is commonly used is Propionibacterium shermani. Bifidobacteria, they actually do not belong to the group of lactic acid bacteria, but they are finding great significance nowadays as starter bacteria because they have a probiotic effect. Probiotic effect means it is a beneficial effect on the health of the host when ingested. These are non-spore forming, gram-positive, catalase negative, non-motile rods. The common examples are Bifidobifidum and Bifidolongum. The next under lactic acid bacteria and related genera is Brevibacteria. A characteristic feature is that it exhibits pleomorphism. Pleomorphism means it exists in more than one form. For example, the young culture can be cocci in shape, whereas as the culture turns old, the cocci are converted into rods. The optimum growth temperature is from 20 to 25 degrees C. It is proteolytic in nature, hence it is used in flavor production in case of some cheese. And the common species which you have is Brevibacterium linens. Now we come over to next group of bacteria which are known as coliform. The term coliform comes since they are associated with the colon, or they are found in the colon. These are gram-negative, non-spore forming cocobacillary rods which are capable of producing lactic acid and gas. They are capable of growing at temperature 30 to 37 degrees C. Why they are significant? Because they indicate post-pasteurization contamination. Because these bacteria are very heat labile and they should get destroyed in pasteurization, but if present in the food product after pasteurization, this means the poor hygienic conditions were prevailing during the processing. The defects which are related to these bacteria are excessive gas production, and they even cause mastitis in milch animals. The common species are Escherichia coli, which is also known as E. coli, and Enterobacter aerogenes. Now we are coming over to a group of bacteria which have much significance in dairy industry. These are the spore formers. Why are they so significant? Because most of them are thermophilic and they survive the heat treatments and later on lead to defects in various milk and milk products. First of all, we will take up the bacilli. These are gram-positive, aerobic, sporogenous rods. Majority of them are thermophilic like earlier I said. Proteolytic in nature, therefore they are associated with various spoilage or defects in dairy products such as bitty cream, Some of them are pathogenic, for example, Bacillus cereus. The next category under spore-forming bacteria is the Clostridia. They differ from Bacillus since they are anaerobic in nature. They are also known as butyric acid bacteria. They are found in soil, plants and manures. They are anaerobes capable of forming spores. They are major spoiler of cheese and they are pathogenic. For example, Clostridium botulinum, this is associated with neural intoxication. The last group of bacteria which is associated with milk is Pseudomonas and related genera. These are gram-negative, motile, aerobic, non-spore forming rods. Why are they so significant in dairy industry? Because they are capable of producing enzymes which are heat stable. These enzymes are lipases and proteinases, because these bacteria, they use fat and protein as energy source. Therefore, they are associated with proteolytic and lipolytic defects in dairy products. The drawback, since they are psychotrophs, they grow during the refrigerated storage and lead to spoilage. The common species which we encounter in dairy industry are Pseudomonas fragi and Pseudomonas putrefaciens. Other related genera are Flavobacterium, Acinetobacter, Chromobacter, Alcaligenes, Aeromonas. Now we are coming over to the next group of microorganisms, that is the fungi. This is a very wide group which comprises of yeast and molds. When we talk about yeast, these are single cell eukaryotic organisms. The shape can be spherical, ellipsoidal or cylindrical in shape. They reproduce by budding or sexually by forming spores. They grow at a temperature between 20 to 30 degrees C and the pH ranges lesser than bacteria. So we can say they grow in acidic environments. So the pH is normally between 3 to 6. These are facultative anaerobes. Since we are talking about yeast, not all the yeast are useful, so we can categorize them into two categories. One is the desirable yeast and other is undesirable yeast. The desirable yeast are the ones which are capable of fermenting or producing alcohol. And the species which are used for alcohol production or alcoholic fermentation are Chloromyces, Saccharomyces and Candida. On the other hand, when we talk about the undesirable yeast, these are the ones which are responsible for the spoilage and defects such as coloration, gasiness, abnormal flavors. Then we come over to the next group of microorganisms which are there in the fungi. These are known as molds. Molds are also known as thread-like fungi due to their structure or their morphology. They are a mass of white cotton-like mycelium which is made up of hyphae. Hyphae are the filaments of the mycelium, you can say, and once we talk about the hyphae, they can be also used as a unit for characterization, because some of them are septate and some of them are aseptate. These molds, they reproduce by variety of spores which can be asexual as well as sexual. The growth temperature of the mold ranges from 20 to 30 degrees C and the pH ranges from 3 to 8.5. Now, molds are also beneficial as well as harmful. When we talk about the desirable molds, some of them are used as starters and some of them are used for the production of microbial rennet. When we talk about their starters, they are used in the cheese industries for the production of white cheeses or blue cheeses. When we talk about the white cheeses, they are produced by using white molds, and a common example is Penicillium camemberti. Hence, this cheese is also known as camembert cheese. On the other hand, when we talk about the blue molds, they produce the blue cheeses, and the common example of a blue mold is Penicillium roqueforti. Therefore, the blue cheese is also known as the roqueforti. Molds, they also bring about some undesirable changes such as discoloration, and button formation, which is commonly seen in condensed milk. The genera which are associated with the spoilage are Alternaria, Aspergillus, Candida, Mucor. Molds are also capable of causing food poisoning because they are capable of producing heat-stable mycotoxins. The most lethal known is the aflatoxin, which is produced by Aspergillus flavus. Then we come over to the last group of microorganisms associated with the milk, that is the viruses. Viruses are acellular. They are obligate intracellular parasites. They are very small in size, therefore they can be only seen by the electron microscope. And their classification is based on their host type. They have lots of public health significance because they are associated with various diseases. As a student of dairy microbiology, the viruses of concern for us are the bacteriophages. These are the viruses which are capable of eating bacteria. They are a big hazard to dairy industry because they lead to the starter failure. Since we have seen what are the microorganisms which are present in milk, and one of the category was of pathogenic microorganism, therefore milk takes a public health significance because it is one food which is consumed by everyone in direct form or indirect form. So we are starting with the next unit, that is milk in relation to public health. When we talk about milk, it is a potential carrier of pathogens. Why? Since milk, we are saying, is a very good source of nutrients, so it will be a very good source of nutrients for the pathogens also. When we talk about milk in relation to pathogens, we come across two terms. One is pathogenicity and other is virulence. When we talk about pathogenicity, it is the ability to cause disease, and the degree of pathogenicity is called the virulence. Now when we talk about a pathogen, it will either invade the human body or it will produce the harmful metabolites. When we talk about these harmful metabolites, these are the toxins. Now these toxins are normally secreted outside the cell or inside. So this leads to their classification on their site of production. So we can have some endotoxins or exo. Exo means outside, endo means inside the cell. The next classification what we have is on their site of action. They can be termed as neurotoxins, means the toxins which are affecting your nervous system. Then we have the cytotoxins which are affecting the vegetative cells or the cells, and one more and very important category of toxin, that is the enterotoxins, means the ones which are attacking the entron. We will be starting with common bacterial pathogens. The first one which falls under this category is Bacillus anthracis. So when we talk about this bacterium, since Bacillus, this means it's a rod which is capable of forming spores, like we have earlier seen the Bacillus group. This is capable of causing a disease that is known as anthrax. The contamination can be traced to the animal root, air and soil. To control this bacteria, there should be thorough cooking of animal products, and steps should be taken to reject milk and meat from infected animals. Most commonly encountered Bacillus species is Bacillus cereus. These are large cell, gram-positive, motile, facultative anaerobes. They lead to food poisoning by enterotoxin. Then we have Brucella abortus. This is responsible for a disease which is known as brucellosis, and it is also spread by direct contact with infected animals. Then we have Campylobacter jejuni. This is an emerging pathogen of concern. It is a slender spirally curved rod which causes a disease, a type of gastroenteritis which is known as campylobacteriosis. Clostridium botulinum, very known pathogen. Although it is rarely found, but the lethality is so high that it is well recorded in the literature. Then we have Clostridium perfringens. This is also an anaerobic spore former which leads to gastroenteritis, and we can easily control this bacteria by quick cooling of heat processed foods. Then we have Coxiella burnetii. This is a causative microbe for Q fever, and this is much more resistant than Mycobacterium tuberculosis. So there are some work going on which are saying that it should replace Mycobacterium tuberculosis as index of pasteurization. E. coli, the most well-known microorganism. It is a rod-shaped facultative anaerobic bacteria which is commonly found in gastrointestinal tracts of humans, hands of handlers, water, sewage. It is a good indicator of post-pasteurization contamination. Strain of E. coli, that is E. coli 0157H7, is an emerging pathogenic strain. We have Listeria monocytogenes. This is also responsible for certain outbreaks associated with milk and milk products. Mycobacterium tuberculosis is a bacteria which is acid-fast in nature and currently used as an index of pasteurization. Salmonella, it is a gram-negative rod which is capable of causing diseases like typhoid fever, salmonellosis. Another member of family Enterobacteriaceae is Shigella. This is capable of causing disease, that is bacillary dysentery. It comes through contaminated food and milk, water. Then we have Staphylococcus aureus. This is a bunch of cocci which usually comes from the hands of the handler, since this is found in the nasal cavity. And if good hygienic practices are not followed, it leads to certain kind of gastroenteritis which are termed as enterotoxicosis. Now we are coming over to fungal pathogens. Although the fungus, most of the fungi are killed by heat, but their toxins, they are left behind because they are heat stable, and they lead to certain defects such as teratogenic effects, and the most studied toxins are the aflatoxins which are produced by Aspergillus flavus and related species. Even viruses are found in milk. The common example are that of Norwalk virus. It leads to viral gastroenteritis. Then we have certain rotaviruses which are there. They also lead to certain kind of diseases if consumed, like infantile diarrhea, winter diarrhea. Hepatitis virus belongs to enterovirus group, and it is also transmitted through various food products and water and ice drinks. Arboviruses is a group of bacteria which is spread through the vectors, insect vectors usually, and it leads to a biphasic milk fever when consumed. So once we have seen what are the various pathogens in milk, now we are coming over to factors which affect the growth of microorganisms. When you talk about the factors, they can be categorized into two categories. The first one is the nutritional factors, and the other category is that of physical factors. So coming over to nutritional factors first. Nutrition is a basic requirement for growth. If there is nutrition, then only the bacteria will grow. So bacterial growth or microbial growth can be defined as orderly increase in the quantity of cellular constituents. In bacteria, it takes place through binary fission. And most of the bacteria, they double, or the population doubles in a specific time period that is known as the generation time or GT. Please refer to table 14.1 in your course contents. You will get the details. The bacterial growth follows a typical growth curve which moves through 4 phases. First is the lag phase, followed by the exponential phase, then the stationary, and finally the death phase. Lag phase, they are trying to adjust themselves to a new environment. Once they've adjusted, they multiply in an exponential manner. Hence, log phase is also known as exponential phase. But this leads to accumulation of toxic metabolites and lack of nutrition, which leads to a plateau stage, which is known as the stationary stage. And finally, this accumulation of toxic metabolites leads to death of these bacteria, that is the death phase. Now, when you talk about the nutritional requirements of cell, for growing they require certain components or substances. These are known as the nutrients. Depending upon the requirement, these nutrients can be categorized into major elements and trace elements. When you talk about major elements, they are required in large amounts. When you talk about trace elements, since the name is trace, this means they are required in very low or minimum quantities. The trace elements, the common ones are magnesium, cobalt, zinc, copper, molybdenum. Now, when the bacteria are growing, they will either utilize a carbon and energy source for their growth. So, on the basis of their carbon and energy source, they are categorized into various categories like photoautotrophs, heterotrophs, chemoautotrophs. Depending if they require light, then they are photoautotrophs. If they require inorganic, they are chemoautotrophs. Once they are growing, the bacteria, they require certain growth factors. Growth factors are very essential because they are not produced by the microorganisms, so they have to be supplied from outside, and the requirement is in very small amounts. Growth factors which are commonly required are purines and pyrimidines. This is for the synthesis of nucleic acid. Then we have the amino acids which are required for the synthesis of proteins, and lastly the vitamins. The detailed account of the vitamins can be referred to table 14.4 in your literature. Now you have the nutrients. So now the bacteria requires to grow. The media which you provide, the medium what you give is the cultural medium. Large bacteria which are there, the medias can be of two categories. One is the liquid media and other is the solid media. Solid media are used for the growth of pure batch cultures, whereas solid media are required for the isolation of cells. Now these culture media, they can be categorized into various categories depending upon their composition. They can be a synthetic media, they can be a complex media. When you talk about a synthetic media, it is made up of an exact known chemical composition. When you talk about a complex media, it is made up of components whose exact composition is not known. Now, depending upon the principles of selection and enrichment, medias can be either selective medias and differential media. Selective, as the name suggests, means they are going to select or they are going to promote the growth of a certain group of bacteria, whereas a differential media is going to differentiate between two sets of microorganisms. The most common example is that of your EMB agar. An enrichment media is basically used for the isolation of pathogens from various samples. This contains a certain component that will permit the growth of specific types of bacteria. When you talk about the physical and the environmental requirements for microbial growth, there are various parameters which are needed. The first parameter which we are going to discuss today is oxygen. And when you talk about oxygen, microorganisms display a wide range of responses to molecular oxygen. And depending upon the requirements, oxygen requirement, they are categorized into various groups such as obligate aerobes, microaerophilic group, obligate anaerobes, facultative anaerobes, and lastly, aerotorrent anaerobes. When you talk about the obligate aerobes, as the name suggests, they will grow only in the presence of oxygen. Whereas microaerophilic, they can grow if the level of the oxygen is not too high. Whereas obligate anaerobes, they will never grow in the presence of oxygen. A common example of aerobe is the bacillus species, which we have just seen, and anaerobe, the common example of obligate anaerobe is the Clostridium. Then we come over to the pH. Once you make the media, the pH is a very crucial factor because all the bacteria, they grow at their optimum pH. They will not grow at the minimum pH, they will not grow at the maximum pH. Microorganisms which grow at an optimum pH well below 7 are known as the acidophiles. The ones which love the neutral pH are the neutrophiles, whereas the ones which grow best under alkaline conditions are called as alkaliphiles. Apart from oxygen, pH, temperature is very crucial for the growth of microorganism, and temperature is one factor which, if taken care into, will also act as a method of preservation, which we are going to see later. So when you talk about the temperature, bacteria, they will grow at various growth temperatures. The ones which grow at low temperatures are known as the psychrophiles or the psychotrophs, whereas the one which love 30 to 37 degrees C, they are known as the mesophiles. And lastly, thermophiles. As the name suggests, therm loving means temperature or high heat loving. So they grow at very high temperatures. Now when you talk about the water, water is very crucial for the growth of microorganism, and availability of water is important for all the microorganisms, and when you see, this is known as water activity. Water activity, which is responsible for the growth of microorganism, ranges from 1 to 0.7. For example, spirillum will grow at water activity which is 1, whereas Helococcus will grow at water activity which is 0.7. The next factor which comes into the play is the osmotic pressure. Since we are talking about the microorganisms, how the changes in pressure, osmotic pressure, will affect their growth. So depending upon the presence of NaCl or sugar in the media, some microorganisms will be inhibited, whereas some will grow. The ones which are going to grow in presence of salt are known as halophiles, and the ones which are going to grow in the presence of sugars are known as osmophiles. When we talk about the halophiles, they can either grow in low concentrations or moderate concentration or even very high concentrations, which range from as low as 1% to as high as 30%. Now we are coming over to the last unit, that is control of microbial spoilage. We have seen the type of microorganisms which were present in the milk. We have also seen the factors which promote their growth. Now we Now we are coming over to Unit 15, that is control of microbial spoilage. Up till now we have seen the type of microorganisms found in milk. We have also seen the factors which affect their growth, and now we are coming over to the final unit which will take care of this microbial spoilage. Spoilage is a deterioration in texture, color, odor or flavor which makes it unsuitable for human consumption. Microbial spoilage of food involves degradation of proteins, carbohydrates, fats by microorganisms or their enzymes. Whenever we talk about milk, we have to make sure that milk should not get contaminated before processing and it should not get contaminated after processing, because when milk comes out from the udder it is nearly sterile. So therefore first aim should be clean milk production, and the target should be milk with lesser or fewer number of microorganisms. And how this can be achieved? We can do it by gentle and complete milking. We can do it by washing of teats and udder with disinfectant solution. Then we can make use of good quality potable water for cleaning purposes. We can utilize ABW system, that is acid boiling water system, for cleaning purposes, and we should take precautions that the cleaning of equipment and containers is up to the mark. Once the milk is stored in the industry, there are chances of formation of biofilms which act as a storehouse of microbes and which lead to further contamination of the incoming milk supply. Therefore, whenever you talk about microbial adhesion, we we were discussing about the unit 15, that was control of microbial growth. There was a technical snag, so we'll continue it further. So when we talk about the control of microbial growth, we can follow the conventional methods which are pertaining to the heat treatment. The most common is the pasteurization, but with the changing time, certain novel techniques are also coming into use, which are like pulse, electric fields, oscillating magnetic fields, and we are having a new concept, that is of Huddle technology. All the techniques have been discussed in detail in your literature, which has been given to you. So before summing up this presentation, I like to conclude that we can control the growth of microbes, and we can also make use of the microbes which are present in the milk for beneficial purposes. And to control them, we should also know the factors which affect their growth. Thank you so much.